everybody. In this video lecture, we continue covering somatic sensory division, special senses, and we will talk about physiology, but this is the second video about physiology of sensation. I think I recorded like 4 or 5 videos already. Seems like it's a very long chapter. So if you're watching it, thank you a lot. Okay, so I'm gonna click over here. So that's my physiology part, and I already covered gustation and audition, that is your hearing, and also equilibrium. So here we are in the vision. Okay, so let's go ahead and begin. And of course, I want to remind you that you need to watch anatomy part first before you start learning about physiology of vision. Now I will go pick up my pen. So vision is your special sensation. You have your special organ, your eye, for this type of information, for visual information. Inside your eye, you have a neural structure that is called retina. And within this retina, you have your receptors, photoreceptors. Now, photoreceptors for visions are rods and cones, so shown over here. This is your receptors shown on the diagram. So that's a rod shown in purple and cone shown in yellow. Now, this cell over here is epithelial cells called a layer that is made of this epithelial cells called a pigmented layer or pigmented epithelium. And you have melanin granules that make it look darker. And embedded inside those cells are your rods and cones. Now they have outer segment. So that's an outer segment, inner segment right here. Then outer fiber, cell body, where you have nucleus and most of the organelles, and inner fibers right there. If you look at the outer segment, you can see those lines. Those lines represent membranous discs. And on our next slide, we're gonna look inside those discs. So here, again, here's your rod. So this is your outer segment, right? And you have your discs. So if you look over here, they took several of those discs and magnified, or not magnified because it's not really histology slide, it's just a picture. So they just draw it bigger. Now embedded in this membrane is the pigment molecule that is called rhodopsin. So here's the name. Rhodopsin. Now those lighter blue part over here are rhodopsin molecules inside the disc. Now if you look at rhodopsin molecule, you will see it's made of what's shown here in the blue part, that is protein called opsin. So here's your opsin. And associated with the opsin is cofactor over here shown in this purple color, and this is retinol. So retinol is cofactor for opsin pigment. That means opsin cannot work without retinol. Retinol is related to vitamin A, and photons, that means your light, activate retinol, right? Now, if you look at the retinol, that is this shown here in the purple color, it actually can have 2 forms. One is shown over here, and you can see that this tail is bended, so it's not straight. And this is called 11-cis retinol. But retinol can also be in all trans form, shown over here. So all trans form when this tail is straight. And this is important Because when light or photon strikes retinol, then retinol changes its shape from 11 cis retinol. It becomes all trans retinol. So from cis, it became trans. So here's our cis, this is our trans. So like I'm in trance. When that happened, the retinol and opsin separate. You see, it goes one way, this goes another way. This is called bleaching of the pigment. So what is bleaching of the pigment? It's like pigment became bleached, right? So, you know, it lost something. It lost this color part, right? I think it's only color here for our better understanding. But bleaching means opsin and retinol split, right? And when it happened, when you have light. So when energy of photon changes the shape of retinol, instead of cis, it became trans, and we call it bleaching of pigment because your pigment, that is your rhodopsin, that's your pigment. Now it loses this retinol and it's only, you know, now it's opsin. So opsin and retinol separate with a bleaching of pigment. Then they reunite again, but you need energy. You need ATP to do it. So ATP energy is used to transform trans back into cis form, and cis and trans form reassemble to form rhodopsin. Now, in the darkness, when you do not see stuff, then your rhodopsin, you know, you have your rhodopsin, and rhodopsin means it's a retinol and opsin together. So you have your rhodopsin, retinol and opsin, and retinol in a cis form. So when it's dark, this is how it exists. When photon strikes, we have bleaching of pigment or separation of trans, retinol, and opsin. So again, this will be important in a minute. But let's see what we have in this PowerPoint. So double bonded carbon change from cis to trans. Yeah, when photon strikes rhodopsin, then this double change carbon, that is a retinol, changes from cis to trans, and this initiate your visual transduction. And visual transduction is the transduction of this visual information from your retina to your brain. Retinol and opsin separate, and that's what bleaching of pigment is. So now let's look over here, what's going on. So we, again, inside those discs, in the outer segment of photoreceptors. So here's the membrane, because it's a cell, it has double membrane. So here's our visual pigment, that is rhodopsin, and here's our cis retinol. Now, when light strikes, we already know that cis became all trans. So now it's straight. That's a bleaching of the pigment. When we have bleaching of the pigment, this actually activates a protein shown here in blue. This is transducin or G protein. So it's activate G protein. Now, when G protein is activated, it activates another protein that called PDE or phosphodiesterase. So it activates these phosphodiesterase, PDE, and this is enzyme. Now, what this enzyme does, it converts cGMP or cyclic GMP into GMP. And GMP is a molecule related to ATP and ADP, right? So it's a nucleotide. So you have this, a lot of this cyclic it has a circular structure, GMP, and it's became just a GMP. And you would think, what a big deal, you know, CGMP became GMP, now what? But the point here is when you have a lot of this CGMP, it's attached itself to the proteins that allow sodium in. Right now, when light strikes, we don't have this CGMP anymore. So this gated channel, sodium and calcium gated channel, it closes, right? Zips up. And sodium and calcium cannot move inside. When sodium cannot move inside, we have hyperpolarization of a cell. Remember, cell is polarized, so it's negative inside. Well, now you prevent positive particles to move in, so you have it, you make it even more negative. So that's what hyperpolarization is. Now, this is gonna be important in the next slide. So let's look at the next slide. So now everything will, you know, make sense for us. So let's look at your retina in dark. So over here in the dark. So what do you have in the retina? So the pigmented epithelium is not shown over here, but here's your receptor, that's your rod. A receptor synapse with bipolar cell, and bipolar cells synapse with ganglion cells. And ganglion cells have these fibers that make optic nerve, right? So that's your optic nerve that delivers this information to your brain, so you can see stuff. But in the dark, you don't see stuff. So now what's going on? Now, actually, in the dark, we have CGMP gated channels that are open, right? So let me remind you, in the dark over here, so when the light strikes, you close the channel. But so just remember here, you see, when the light strikes, what happened? The light closes the channel. So when we don't have light, then the channels are open, right? That makes sense. So channels open, and it's shown over here. Your channels are open because it's dark. Sodium and calcium moves in, and it causes depolarization, because if cells are negative inside and you move positive inside, that's called depolarization. And guess what? This receptor cell is generating receptor potential. So it starts releasing neurotransmitters over here. But the point is, this neurotransmitter is inhibitory. It's not excitatory. And what it does, it inhibits your bipolar cells. And when bipolar cells are inhibited, they cannot release their neurotransmitters. They cannot excite your ganglion cells. That means there is no action potential moved to your brain. No, right? So in the dark, actually your photoreceptors are busy. In the dark, you would think that should be opposite way, but no, this is how you work. When you rest, when you close your eyes, when you don't see anything, your photoreceptors are actually very active. They releasing neurotransmitter, but this neurotransmitter inhibits everything else, and you do not see stuff. So no action potential occurs along the optic nerve. That means there is no impulse that would reach your brain. Now, what happened in the light? In the light, let's go back to previous slide over here. In the light, when you have bleaching of pigment, when you activate G protein, and G protein activate phosphodiesterase, then you don't have this CGMP anymore and channels are closed. So sodium and calcium cannot move in, right? So it's shown over here. In the dark, when light strikes, so here light strikes, now CGMP gated channels now are closed. So now your receptor cells are pretty much resting. They stop releasing neurotransmitters, because if sodium cannot go in, right, we cannot, then the calcium will not go in, and we cannot release neurotransmitter. So when neurotransmitter is not released by your receptor cells, then guess what? Bipolar cells now happy. Now they are not inhibited. They start to release neurotransmitters. They activate ganglion cells, and ganglion cells create action potential along the optic nerve. They will travel to your brain, activate your primary visual cortex, and you will see stuff around you. So, right? So just remember, in the dark, your photoreceptors are working, they're releasing neurotransmitter, but though this neurotransmitter is inhibitory, so everything is inhibited and signal is not reaching your cortex. In the light, your photoreceptors now inhibited, your photoreceptors inhibited, so they don't release neurotransmitter, then everything else is activated by polar ganglion cells and optic nerve, and you send signal to your brain. That's amazing how our body works. Sometimes you would never guess it would be this way. If I never studied it and they would told me, make your prediction how it works, I would probably predict something completely different mechanism, but this is how it is. Okay, so what we cover the vision, right? Phototransduction pathway. We also mentioned bleaching of the pigment that actually starts this phototransduction. Now we will talk about briefly, about central processing. So not only you need to excite and activate your receptor cells, right? You also need to send this signal to the brain. So let's see how that happened. Central processing through the spinal cord. So some sensory information, of course, will go through your spinal cord to your brain, and other information will go straight to your brain. Now, this diagram show you central processing through spinal cord and then to the brain. Now, in a spinal cord, that is actually, that's already your medulla. So here, just this part and this part, that's your spinal cord. You have pathway for your sensory information, and it's ascending pathway. Ascending means you're moving towards the brain. So ascending pathway carries sensory information to the brain. And there is several of this pathway. One of them called dorsal column. So dorsal means to the back, right? So here on the back, over here and here, you have this dorsal column. And this is one way to carry information. And dorsal column carry information of touch, vibration, touch, vibration, pressure, and proprioception, right? That's the dorsal column. Pressure, I'm not sure. Let's just say touch and proprioception. So this is the dorsal column. If somebody touching you, that's where this action potential electrical impulse moves. So it moves from spinal cord to your medulla, that is a part of the brain, to the midbrain. From midbrain, it moves to the thalamus, and then thalamus directs this information to a specific area of your cerebral cortex. So that's your cerebrum. It's the biggest part of your brain, and on the surface, you have this cerebral cortex. So the thalamus is going to send information to your cerebral cortex, any place it needs to go. And information that reaches your cerebral cortex reaches your consciousness. So you understand this information. There is another pathway. It's another tract, right? So we have this, dorsal column, and we have a spinal thalamic tract. So this is where it's located. It's a lateral. We also have kind of like the we have several spinothalamic tracts, but lateral is more studied. Always they give us example of lateral spinothalamic tract. Guess why? Because it carries very important information about pain and temperature. So now if somebody hits you, I hope not, but if you cut yourself, or temperature, this information will be carried through the spinal thalamic. So it's spinal means from spinal cord, thalamic means to the thalamus. So here we move through spinal thalamic tract. Again, we passing medulla, midbrain, and then we move to the thalamus, and from thalamus to the cerebral cortex. Now, we are not going into detail over here about all this first and second and third order neurons. This is for nervous system lecture, right? But so far, I hope it makes sense. So not only you need your receptors to sense the stimulus, sense touch or pain or temperature, you also need those receptors to send signal to your cerebral cortex, so it can reach your brain. It can you can understand the sensation. Now, this picture show you the cerebral cortex over here, and your sensory information, general sensation. Remember, sensory information can be general senses and special senses. General senses, you don't have a special organ for this sensation, so it comes from your skin, from all over your body. And special senses, you have your like special organ, like eye, nose, tongue. And even if you see here eye, nose, and lips and tongue, this is not information about taste or vision or smell. This is when somebody touches your eye. This is when your nose is cold, or this is when you eat ice cream and you feel like how cold it is. That's a sensory information. It's a general sensory, not specific sensory. So this general sensory information is received by your parietal lobe, right? So you have your frontal lobe and you have your parietal lobe. And in the parietal lobe, you have this somatosensory cortex, and information from different parts of your body by thalamus sends this information to a different part of your cortex, right? For example, food, toes, and genitals, the information will come more medial, right? And more lateral, you have your information from fingers, from hands. This diagram is called sensory homunculus. Sensory homunculus is just like a person, like very weird looking person, that show you where exactly information travels from your sensory receptors. And you can see like hand is so huge and fingers are so huge compared to toes. That means you have way more sensory receptors in your fingers, and the larger area of the cortex dedicated sensation from your fingers. Now look, how big your tongue is. That means lots of sensory information come from your tongue, right? Lots of information come from your lips here and here, from nose, from eyes, and very little comes, for example, from for example, from your foot, you have way less information, right? From your hip. Over here, you, by the way, have neck. Right, so that's a sensory homunculus, and this is for somatosensory receptors. Right, so if you go back to this slide, you guys can see, so for example, you have touch, or you have, you feel temperature from your hand, from your toes, from your nose, from your lips, and this sensory information is sent to your cerebral cortex, but it's not sent like anywhere, right? It's sent into a precise area of the cortex, and when we use this homunculus, we can see where exactly this information goes. Next. So the information that we just discussed was somatosensory information that travels from periphery, of your body to your spinal cord, to your brain, and to your cerebral cortex. But we have different pathways. For example, trigeminal pathway. You have a cranial nerve. This is cranial nerve number 5, called trigeminal nerve. Now, the trigeminal nerve is cranial nerve. That means it connects to your brain. It doesn't connect to your spinal cord, so your information doesn't go to your spinal cord. So it's somatosensory information from face, head, mouth, nasal cavities, right? So over here, so show you trigeminal has 3 branches. So this sensory information travels from trigeminal nerve straight to your brain without going to the spinal cord first. So axons or nerves project to one of the 3 locations: spinal trigeminal nucleus, chief sensory nucleus, mesencephalic nucleus. That we're not gonna look at this location because you need to study nervous system to understand where this area is located. But it goes to somatosensory cortex in the cerebrum after that. This is the one that I showed you before, that I'm going to show you again. It goes to this somatosensory cortex, right? So your nose, lips, blah, blah, blah. So over here, you skip your spinal cord, right? Because you go straight to the brain and then to the cerebral cortex. This diagram shows you the condition that is called trigeminal neuroglia. Here's the name. And this is really very, very severe, sharp pain in the face. Symptoms showed up periodically, and in some cases, it seems like a toothache. But this is extremely painful condition. You can see it can cause what is this? So it can be caused by chewing, weather, stress, even tilting your head. They call it suicidal pain. In the everyday language, because it's very, very famous, very, very painful sensation, and it caused by some lesions or some damage of the trigeminal nerve. It occurs when a blood vessel moves too close to the 5th cranial nerve, also known as trigeminal nerve, and nerve become inflamed. Right, so when you have inflammation of the nerve, then you might have this really bad pain. Now let's look at our special senses, because what we discussed before, information that is received by your somatosensory cortex, that's a general sensation. Special sensation might have some, different pathway. Gustation. We do talk about the taste, right? How we have this taste buds, how we have receptors here that are activated by different chemical molecules, and we look at activation by sugar molecule, for example. But when the receptors are activated, they activate those nerve fibers, and nerve fibers carry action potential towards your medulla oblongata, then pons, that's a part of brain. So straight to your brain, thalamus, and then a gustatory cortex. And gustatory cortex found in a lobe of the cerebrum that's called insula. That's right there. And those nerves that carry taste to your insula can be glossopharyngeal, facial or vagus nerve. Right, so that's why we call it sensory processing. We want to describe how information is received by your brain, right? So from your tongue through cranial nerve to your brain, including your thalamus, that is a part of your brain, and then to the cortex of insula. Auditory pathway. So we also already looked at our audition. So here's our cochlea, right? So this is our cochlea duct. Here's organ of Corti, or sensory organ. Here's our hair cells. When these hair cells excite, they excite the bipolar cells of the cochlear nerve. Then cochlear nerve became vestibular cochlear nerve, or nerve number 8, and information moves to the medulla. And again, this is the brain. So medulla, midbrain, and thalamus. And thalamus sends this information into primary auditory cortex that is located in temporal bone. All right, so that was auditory pathway. Now, location of sound determined by comparing information from both ears, and your ears look at a time difference, so when a sound reaches right and left ear, and intensity difference, so where the sound is louder, and comparing these two factors, you can determine where the source of sound is. Now, balance pathway. So here's again our inner ear. That's a cochlea that is involved in hearing, but now we're looking at vestibule and semicircular canals. Now we have our hair cells over there. When hair cells are activated, they activate a nerve, and this is vestibular branch of vestibular cochlear nerve. So here's the branch, and then they became one structure, and we move to the brain. So we're not going through all these different parts. We know we move to the medulla, pons, right? That's the brain, of course, midbrain, and thalamus again, and from thalamus to the cortex, and this is also in your temporal area over here. Right. Actually, all your sensory information, with exception of olfaction or smell, move to the thalamus, and then thalamus directs it into appropriate part of the cerebral cortex. So that's a balance. Now, vestibular ocular reflex allows for stable vision upon head movement. So eye movements in opposite direction of head in 1:1 ratio. So if you look on a picture here, so this is our semicircular canals, and some nerve fibers from semicircular canals. So we just look at previously, let's look previously from semicircular canal. You see how they go to the medulla, pons, thalamus, right, and cerebral cortex. But some of them are actually, some of these fibers, those are motor fibers now. Previously, we look at sensory fibers, but some motor fibers, they move and innervate your eye muscles, like lateral rectus, medial rectus shows over here, and they either inhibit or excite those muscles, and those muscles move your eyeballs in a direction opposite to the direction of your head. And vision pathway, probably the most complicated pathway over here. Now, so here's our left side, this is our right side, and you can see that informational light. So here's your retina. So when light strikes your retina, you have your photoreceptors that are excited, and they send, well, they actually, remember, photoreceptors, they get inhibited, and that's why your bipolar cells are excited and ganglion cells are excited. And so the impulse is sent to your brain. specifically, of course, to the cortex, specifically in the occipital lobe. So here's you have your primary visual cortex in the occipital lobe. When you look at your retina, so your nose would be here. So this part of the retina is called nasal retina. And this part of a retina on the lateral side is called temporal retina because you have your temporal lobe over here. Now the light that strikes, so that information that falls on nasal retina, look over here. So this green one right there, and this purple over here, they cross. So information from here kind of move to the opposite side, and information from over here moves to the opposite side. But the information that strike temporal retina, it does not cross. Right, so that's very important, and it has some clinical applications that we will look at in a moment. What we can see here, so from the retina, the information it passed through the what we call optic nerve, right? So that's a retina, then those fibers are called optic nerves. Then this structure in the middle is called optic chiasm, and the optic chiasm you have optic tracts. From optic tracts, you have this optic radiation, and you reaches your occipital lobe, that is primary auditory cortex. Now let's see how this crossing of information make significance. Let me see over here. What it says here that I mean is that you have a field of view, right? So this is left visual field, right? So this is left visual field. So what falls on the left visual field? This part and this part. And you can see that this part over here goes right there, and this part over here goes also on the right side. So information from left visual field processed in the right visual cortex, and information from the right visual field, that is this part and this part, shown kind of in green, right? It processed in the left visual cortex. And of course, we have a binocular field in the middle where both hemispheres are involved, right? But again, information from visual field shown in purple. But it's interesting that information from visual field you receive by both eyes, right? You not only receive it by left eye. It's not like you have this line over here and your left eye just sees this part and your right eye sees this part. But that's why we kind of referring to the field, because from left visual field, information shown in purple. So you can follow the purple. So see how, for example, I have something here, and you see it with both eyes, right? So information falls to the temporal retina here and nasal retina. Everything is in the purple is received by right visual cortex. Now, if you have like some object over here, like a little flower, then in the light reflected from the flower shown here in the green color, and you can see it's received by both eyes, right? But the information is processed by left visual cortex. And it became important when you have some lesions in this visual pathway. So, for example, let's look right here. So what if you have, so that's your right eye right here, right? This is right, this is left, right? So keep it in mind. So let me write it with, not with a vanishing pen, but so that's right, and this is left. So now I'll get my vanishing pen. Now, what if we have, so this is my right eye. So this is the retina, this is temporal retina and nasal retina of the right eye. And this is my right optic nerve. So if I have lesion here, then nothing from the right eye reaches my brain. So this is how you would see. So there is no vision from right eye, but left eye is not affected. And it does make sense, you know, right eye is affected. I cannot see anything with right eye, but this is where it gets interesting. So for example, I have lesion over here in the left, not optic nerve, but optic tract. This is optic nerve, this is optic tract. So when you have lesion in the left optic tract, you would think, okay, it's a left optic tract, then no vision in the left eye, but that's not how it is. Because what information is not reaching the cortex over here? Look, this yellow information. So information that comes from here, you cannot see it, right? So you cannot see that one. But information that comes from this side, that's actually fine because it's crosses. What else is not? The information that falls on this side. So this red one, this is not reaching. So for number 4, look, that's how a patient would see. So you will not see, you will not receive any information from the right visual field. That's interesting, right? When the left optic tract is damaged, right, then you will partially see with both eyes, right? You just will not be able to see stuff that is on the right of you, and so on, right? So if you have a damage over here, for example, you can see now this yellow part, it goes through, the blue part goes through, right? So in green and red, what I mean, of course, it's not in green, but information that falls on the nasal part of the retina now cannot reach your cortex. So we cannot see anything from here and over here. That is red. So not this one, no red one. And this is number 2. That's called bitemporal hemianopia. So that's interesting. Of course, I don't expect you to memorize, you know, any of these pathways. Just remember that kind of whatever is lateral, your temporal retina, these fibers do not cross, and fibers from nasal retina, they cross. Okay, and I think this is my last slide. Yes. Okay. So finally, I finished this chapter. I recorded 5 or 6 videos, and I really appreciate you guys if you watch it till the end. I doubt. I just wonder if any single person watched this video till the very end. Please write me a comment. Yes, I watched it till the end, or don't write anything, because of course you will not hear what I just said, right? But thank you so much for watching. I hope it was helpful, and I hope it all make sense for you. I'll talk to you next time in my next video lecture.